medcram.com.
Welcome to another MedCram COVID-19 update, and we're going to be talking about the AstraZeneca vaccine in this update. But first, let's take a look at the numbers real quick. Daily new cases in the United States are at an all-time high, but it may be peaking. We don't know. We'll need to get some more data here. But if you look at the data, you can see that there has been somewhat of a slowdown in the number of cases. Of course, with deaths, they lag behind maybe a couple to 3 weeks, and the daily new deaths are increasing as well at this point. Again, we're going to have to wait for further data. So now we're going to talk about the AstraZeneca Oxford vaccine. And we're going to compare and contrast to the ones that have gone before it, like the Pfizer BioNTech vaccine and the Moderna vaccine. Now, as you will recall, the Pfizer BioNTech vaccine and the Moderna vaccine are basically new vaccines in the sense that they are injecting messenger RNA into your cells to tell the cells to produce the protein that is the same as a portion of the spike protein on the virus, and that is what generates the immune response. So in other words, we're giving instructions to the cell to make the foreign protein which causes the immune response. And that is actually very similar to the AstraZeneca Oxford vaccine, except instead of using messenger RNA, they are using DNA to do that. So once again, in terms of a picture, imagine we have the cell and we've got the nucleus. And what we were doing before was we were taking messenger RNA, and that messenger RNA was being translated by ribosomes and converted into the protein. And what we did with the Pfizer BioNTech vaccine and the Moderna vaccine is we were getting the messenger RNA into the cytoplasm of the cell. The cytoplasm is the outside portion, as opposed to the nucleus. How do we get that messenger RNA in the Pfizer BioNTech vaccine and the Moderna vaccine in? Well, we used a lipid bilayer, or a micelle as we called it, to get those fragments or that messenger RNA into the cytoplasm. And then from there, it's fairly straightforward. The cell uses its own cell machinery, which is the ribosomes, to cause translation to occur, and you make the protein in question. How this is different is AstraZeneca uses a different technique, and it uses a virus called an adenovirus, but not a human one, but a chimpanzee. Now, why would it do that? Chimpanzee adenoviruses have not really been exposed to the human population, and so it's very unlikely that humans would create an immune response against the adenovirus specifically itself. We don't want that to happen, because if that happens, then the vaccine is not going to be effective at getting its instructions into the cell. And of course, AstraZeneca knows that if this vaccine is successful and it's used on a number of people, millions, billions of people worldwide, then it's probably going to have to abandon the chimpanzee adenovirus platform for delivering instructions. Now, the adenovirus here does not use RNA like we've been talking about. It uses DNA. And DNA is a double-stranded form of instructions. Now, there's a portion there in the middle of that is used for replication of the virus. Well, the genetic engineers simply delete that portion so the virus can no longer replicate and cause an infection in the human body. And instead, they replace that portion of the DNA with the genetic code, the instructions that the geneticists have engineered. And so what happens when you deliver this adenovirus in the vaccine, it goes into the cell and it releases the DNA into the cytoplasm. And then that gets taken up and goes into the nucleus. That DNA, according to scientists at AstraZeneca, does not get incorporated into the host's genome. It does not go into your DNA in the cell, but rather it is transcribed into RNA. And that RNA then exits the nucleus as a messenger RNA, and then exactly the same thing happens as what we saw with the Pfizer BioNTech vaccine and the Moderna vaccine, which is that the messenger RNA, which is now a product of the transcription of the DNA, gets converted into the spike protein through translation by the ribosomes, and then this goes on once again to cause the immunogenic response. Now, there's a couple of points that you should understand about the trial, the AstraZeneca and Oxford trial that's been looking at this vaccine. And let me just stress to you that the results that we have gotten here in the last few days are via press releases and not peer-reviewed articles that have been published and reviewed. So the first difference, as we talked about, is that it is a DNA virus platform. And there's been many areas that they've been doing research. And predominantly, let's take a look at two of those areas. The first one was in Brazil. And the second area that they did the trials was in the UK. Be also aware, though, that they are doing trials in the United States as well. And from the numbers, and I'm rounding these off, there's been about 9,000 people that have been enrolled in Brazil and about 3,000 that were enrolled in the UK. Now, just like the other two vaccines that we were talking about, this is also a two-step process. So there's two injections or two vaccinations, and they're about a month apart. Now, by some error that occurred in the UK trial, they gave half a dose first, followed by a full dose. Now, in the Brazil trial, they gave a full dose like they were supposed to, followed by a full dose, and this happened approximately 28 days apart. Now, if you've listened to us when we talked about the other two vaccines, you'll know that in those two vaccines, they did the trial a little differently in terms of the fact that they waited for subjects to become symptomatic with COVID-19 symptoms before looking at the endpoint there, or a midway point, to see whether or not the vaccine was effective. Here in the AstraZeneca case, they were not doing it that way. No, in fact, they were doing swabbing and they were checking for infections. This is a huge deal because whereas with the Pfizer BioNTech vaccine candidate and also the Moderna vaccine, all we can really say about those two, even now with the results that we have, is that it is definitely effective, if what they're saying is true, at preventing the symptoms of COVID-19. At preventing the disease, at preventing the things that cause people to go to the hospital. What we don't know right now about those two vaccines is whether or not the vaccine prevents infections. And that's important because infections leads to transmission, and transmission leads to spread of the virus. Now here, with the AstraZeneca Oxford study, they actually checked for infections by doing nasal swabs. And so what we can say here is whether or not there has been a stoppage or a ceasing of transmission and therefore infections. Now, what was really interesting and kind of a head-scratcher is in the Brazilian portion of this study where they gave the full dose on the first injection and the full dose on the second injection, the efficacy was only about 62%. Now, I say only because we're comparing it to what we know now about the other two vaccines, but 62% is not a bad result, and it certainly meets the benchmark or the threshold for getting emergency use authorization from the FDA, both here in the United States and also in Europe. However, the part that's kind of a head-scratcher is that when they, by some serendipitous error, they injected the subjects in the UK with half a dose on the first shot and then a full dose on the second shot, this one was actually 90% effective. And that's not just for preventing COVID-19, as I said. That may also, according to the preliminary results, prevent infections from what they're telling us. And so the U.S. trial was paused because there were some events in two subjects in the United Kingdom and one subject in Brazil that was deemed later not to be related to the vaccine, but it took them time to investigate. They have restarted the USA portion of the trial, but now they want to see if they can, in midstream, modify that to introduce some half dosing as well, combined with perhaps full dosing, to see whether or not they're going to get a similar result, either 62 or 90% or something in between. Now, the other thing that was very interesting about this is that obviously some people are getting placebos. But in not every situation are those placebos just saline or salt water. In fact, in the first injection, what they had been doing is getting a meningitis vaccine dose because they wanted to reproduce the symptoms that people got when they got an injection. Because when you get an injection with saline, it doesn't cause irritation at the site as much. It doesn't cause pain at the site as much. But a regular vaccine, which would have nothing to do with coronavirus, such as a meningitis vaccine, might do that. And so here, AstraZeneca-Oxford trial is a little different than the other two in that they were giving other vaccines as a placebo. And the reason why they're doing this is to get a more realistic placebo effect so that the subjects can't tell whether they've gotten the vaccine in this case or whether they're in the placebo arm. So what are some other possible advantages? Well, with the Moderna vaccine, as we talked about, it had to be stored, if you wanted to store it for 30 days, at negative 20 degrees centigrade or negative 4 degrees Fahrenheit. And as you recall, for the Pfizer vaccine, it was even more extreme. It was at negative 70 degrees centigrade or negative 94 degrees Fahrenheit to store it for 6 months. Here, just a regular refrigerator is all that is necessary. In fact, it only needs to be stored at 2 to 8 degrees centigrade or 36 to 46 degrees Fahrenheit. So those are some advantages there. And of course, because of that, you don't need to build an infrastructure system. You just need regular refrigeration to store this vaccine. The other thing that's very interesting as well is that AstraZeneca has promised not to make any profit during the pandemic on this vaccine. And so while the cost of giving the other two vaccines is around the $20 range, the AstraZeneca Oxford vaccine is going to be sold for anywhere between $3 to $5. And not only that, the AstraZeneca Oxford vaccine is potentially going to hit about 3 billion doses. And what's interesting is that because they only needed a half dose here for the 90% efficacy, they would actually be able to save some of that material, and that could immediately go as high as 4.5 billion doses worldwide. The other thing that's interesting politically as well is that because the United Kingdom is leaving the EU, they would be able to get this fast-tracked within their own country without depending on EU approval. However, they are applying for EU approval as well. Other things that came out in that press release is that there were no serious side effects. The other thing that said, too, was that there were no severe COVID cases in those patients that got the vaccine. So let's review. How is the AstraZeneca Oxford vaccine similar to the Pfizer BioNTech and the Moderna vaccine? Well, it's similar in that 2 vaccinations are necessary, approximately 1 month apart. It's also similar in that it delivers the instructions to your cell for the cell itself to make the protein antigen that stimulates the body to make the immune response. But this candidate is different in the fact that it's a DNA virus. It's also different that it doesn't really require sub-zero temperatures to preserve it. It's also different in terms of the fact that it's going to be a lot more affordable, especially for countries that don't have the infrastructure to keep these vaccines at very low temperatures. And it's also different that there is this head-scratcher type of situation here, where we don't really understand why this small UK group had such high efficacy, especially with only half the dose on the first dose. So right now, we're kind of feeling around here in the dark. What we really need to have, and what we are going to have very shortly, is the actual data released that will be peer-reviewed and published. And if you want to read more about this, I'm going to put in the description below the links where we got this information from so you can do more research and find out for yourself. If you want to hear more medical topics explained clearly, please visit us at medcram.com. Thanks for joining us.